Hi class, this is your instructor, Skylar Huff. We're now in chapter 9. This is meiosis. So you will distinguish sexual from asexual reproduction, haploid from diploid, and gamete from zygote, and meiosis from mitosis. Only a few years after Walter Fleming's discovery of chromosomes in 1879, Belgian scientist Pierre-Joseph van Beneden was surprised to find different numbers of chromosomes in different types of cells in the roundworm Ascaris. Specifically, he observed that the gametes, eggs and sperm, each contained 2 chromosomes, whereas the somatic or non-reproductive cells of embryos and mature individuals each contained 4. From his observations, van Beneden proposed in 1887 that an egg and a sperm, each containing half the complement of chromosomes found in other cells, fuse to produce a single cell called a zygote. The zygote is, like all of the somatic cells, ultimately derived from it, contains 2 copies of each chromosome. The fusion of gametes to form a new cell is called fertilization, or syngamy. So it was clear even to early investigators that gamete formation, here we go. It was clear even to the early investigators that gamete formation must involve some mechanism that reduces the number of chromosomes to half the number found in other cells. If it did not, the number of chromosomes would double with each fertilization, and after only a few generations, the number of chromosomes in each cell would become impossibly large. For example, after 1 generation, the 46 chromosomes present in human cells would increase to 92 chromosomes. After 2 generations, it would increase to 184, and by 10 generations, the 46 chromosomes present in human cells would increase to over 47,000 chromosomes. Clearly, that was not the case. The number of chromosomes does not explode in this way because of a special reduction in chromosome division that occurs during gamete formation, producing cells with half the number of chromosomes. The subsequent fusion of the 2 cells ensures a consistent chromosome number from 1 generation to the next. This reduction division involves a process known as meiosis. That is the process. So sexual reproduction. Meiosis and fertilization together constitute a cycle of reproduction. 2 sets of chromosomes are present in the somatic cells of adult individuals, making them diploid cells. And diploid is denoted by 2N. That's number 2N. But only 1 set is present in the gametes, which are thus haploid. And haploid is indicated by a singular N, no 1 needed, just the letter N. So 2N, diploid, N, haploid. For example, if a sperm cell containing 3 chromosomes from the father fused with an egg containing 3 chromosomes from the mother, a diploid zygote with 6 chromosomes would be the result. Reproduction that involves this alternation of meiosis and fertilization is called sexual reproduction. Some organisms, however, reproduce by mitotic division and don't involve the fusion of gametes. Reproduction in these organisms is referred to as asexual reproduction. Many plants and some animals are able to reproduce both asexually and sexually. The sexual life cycle. So here you will compare the life cycles of plants and animals, distinguishing between somatic and germline cells. Having 2 parents is the basic to being an animal or plant. The life cycles of all sexual reproducing organisms follow the same basic pattern of alternation between diploid chromosome numbers and haploid ones. In unicellular eukaryotic organisms, like the protist shown in figure 9.4a, that's there to the left, as you see in the bottom left hand of your screen, or the middle left hand portion of your screen, So individuals with haploid for most of their lives. The protist individual. When they encounter environmental stress, haploid cells sometimes fuse with other haploid cells to form a diploid cell. Later, this cell undergoes meiosis to reform the haploid phase. In most animals, like the frog you see in the very same figure, 9.4, however, this is 9.4b, meiosis forms gametes, and fertilization results from the formation of a diploid zygote. This single diploid cell divides by mitosis, eventually giving rise to the adult frog shown in the photo. Almost all animals are diploid for the multicellular stage of their life cycle. In plants, like the fern you see in figure 9.4c, haploid and diploid individuals alternate. Certain cells of a diploid individual undergo meiosis, making haploid cells or gametes that divide repeatedly by mitosis to form a multicellular haploid individual. Some cells of this haploid individual eventually differentiate into eggs or sperm, which fuse to form a diploid zygote. Dividing by mitosis, the zygote forms a diploid individual. In animals, the tissues that will eventually undergo meiosis to produce gametes are set aside from other cells early in the course of development. The cells of the body are somatic cells, while gamete-forming cells, located in the reproductive organs of males and females, are referred to as germline cells. Both the somatic cells and the gamete-producing germline cells are diploid. Somatic cells undergo mitosis to form the genetically identical diploid daughter cells, and then the germline cells undergo meiosis, producing haploid gametes. So here is figure 9.3, which shows sexual life cycles in animals. So this is it. Stages of meiosis. So let's look more closely at the process of meiosis. Just as in mitosis, the chromosomes have replicated before meiosis begins during the period called interphase. The first of the two divisions of meiosis is called meiosis 1, and it serves to separate the homologous chromosomes, or homologs. The second division, or meiosis 2, serves to separate the sister chromatids. Thus, when meiosis is complete, what started out as one diploid cell ends up as 4 haploid cells because there was one replication of DNA but 2 cell divisions. This process reduces chromosome number by half. Meiosis 1. Let's outline what happens in the 4 stages of meiosis 1. So meiosis 1 traditionally is divided into 4 stages. Please see the left side of the essential biological process, 9a, which is shown in your textbook on page 158 and 159. Prophase 1. 2 versions of each chromosome, the 2 homologs, pair up and exchange segments, or I'll say genetic information. Next is metaphase 1. The chromosomes align at the central plane, or metaphase plate. Thirdly, one homolog with its 2 sister chromatids still attached move to a pole of the cell, and the other homologs move to the opposite pole. That is anaphase 1, and then is telophase or telophase 1. Individual chromosomes together gather at 2 poles. So as a review, let's get to the process. Prophase 1. Individual chromosomes first become visible when viewed with light microscope, as our DNA coils more and more tightly. Because the DNA replicates before the onset of meiosis, each of the thread-like chromosomes actually consists of 2 sister chromatids joined at the centromeres, just as in meiosis. However, now meiosis begins to differ from mitosis. And it differs from mitosis in that during prophase 1, the 2 homologous chromosomes line up side by side, physically touching each other, as you see in figure 9.5. At this point, the process known as crossover or crossing over is initiated, in which the chromosomes actually break in the same place on both non-sister chromatids, and sections of chromosomes are swapped between the homologous chromosomes, producing a hybrid chromosome that is part maternal chromosome and part paternal chromosome. This is just one of the ways that meiosis provides us with variation or differences. Up next, we get to what is called metaphase 1. So, to review crossover or crossing over, if you look at the bottom, that is the result of synapsis, which is shown here in the center, where they actually touch each other and they pair up is synapsis, meaning those both could be chromosome 1s. They're both chromosome 2s, meaning that is a green and that is a purple. At the bottom, because of crossing over, they have exchanged genetic information. In metaphase 1, the spindle apparatus forms, but because homologs are held close together by crossovers, spindle fibers can attach to only one outward-facing portion of each centromere. For each pair of homologs, the orientation on the metaphase plate is random. This is a second way that meiosis provides us with variation, which is called independent assortment. Each orientation of homologs results in gametes with different combinations of parental chromosomes. This shows that here. Anaphase 1. The spindle fibers shorten and the homologs are pulled apart and move toward opposite poles. At the end of anaphase 1, each pole has half as many chromosomes as were present in the cell when meiosis began. And remember that chromosomes replicated and thus contain two sister chromatids at the start of meiosis. Well, now the sister chromatids are not counted as separate chromosomes. As in mitosis, count the number of centromeres to determine the number of chromosomes. And lastly, we're now here at telophase 1. So in telophase 1, the chromosomes gather at their respective poles to form two chromosome clusters. After an interval, meiosis 2 occurs, in which the sister chromatids are separated as in mitosis. Next up, meiosis 2. So you will outline the events of meiosis 2, stating how many copies of each chromosome are present at the end. So following meiosis 1, the cell enters a brief interphase in which no DNA synthesis occurs, and then the second meiotic division begins. Meiosis 2 is simply a mitotic division involving the products of meiosis 1. Please see Essential Biological Process 9A, as shown on page 158 and page 159. At the end of telophase 1, each pole has a haploid complement of chromosomes, each of which is still composed of two sister chromatids attached at the centromere. And like meiosis 1, meiosis 2 is divided also into 4 stages. Let's begin with prophase 2. Prophase 2 is quite similar to what is called prophase of mitosis. So at the two poles of the cell, clusters of chromosomes enter a brief prophase 2, where new spindles form. In metaphase 2, the spindle fibers bind to both sides of the centromere, and the chromosomes line up along the central plane or metaphase plate. Anaphase 2 is where the spindle fibers shorten, splitting the chromosomes and moving the sister chromatids to opposite poles. And lastly, the nuclear envelope reforms around the 4 sets of daughter chromosomes. Meiosis 1 is shown here. Meiosis 2 is shown here with 4 genetically different haploid cells. So the main outcome of the 4 stages of meiosis 2 is to separate the sister chromatids. The final result of this division is 4 cells containing haploid sets of chromosomes. No 2 are alike because of the crossing over in prophase 1, independent assortment, and even the segregation that happened in anaphase 1 and anaphase 2. If you think about it, the key to meiosis is that the sister chromatids of each chromosome do not separate from each other in the first division, prevented by the crossing over that occurred in prophase 1. So imagine 2 people dancing closely. You can tie a rope to the back of each person's belt, but if you cannot tie the second rope to the person's belt buckles in front, the 2 chambers are facing each other and are very close. Just in the same way, microtubules cannot attach to the inner sides of centromeres to pull each of the two sister chromatids apart because crossing over holds the homologous chromosomes together like dancing partners. Let's continue on now. So, how meiosis differs from mitosis. So, you will explain two unique features that distinguish meiosis from mitosis. While there are differences between eukaryotes in the details of meiosis, two consistent features are seen in the meiotic processes of every eukaryotic organism or every eukaryote: synapsis and the reduction division. Indeed, these two unique features are the key differences that distinguish meiosis from mitosis, which you studied in Chapter 8. Synapsis. The first of these two features happen only during the first nuclear division. Following chromosome replication, homologous chromosomes or homologous pairs pair all along their lengths. These sister chromatids are held together by proteins called cohesin. While the homologs are thus physically joined, genetic exchange occurs at one or more points between them. The process of forming these complexes of homologs or homologous chromosomes, it is called synapsis, and the exchange process between paired homologs, as described earlier, is crossing over. So figure 9.8a shows how the homologs are held together closely enough that they are able to physically exchange segments of their DNA. Sister chromatids do not separate from one another in each of the nuclear divisions, so each homolog is still composed of two sister chromatids, and they're joined at the centromere and still considered one chromosome. Secondly, the reduction division. The second unique feature of meiosis is that the chromosome homologs do not replicate between the two nuclear divisions, so that chromosome assortment in the second division separates sister chromatids of each chromosome into two daughter cells. In most respects, the second meiotic division is identical to a normal mitotic division. However, because of the crossing over that occurred via the first division, the sister chromatids in meiosis 2 are not identical to each other. Also, there are only about half the number of chromosomes in each cell at the beginning of meiosis 2 because only one of each homolog is present. So figure 9.8b shows the reduction division as it occurs. The diploid cell in the figure contains 4 chromosomes, 2 homologous pairs, and after meiosis 1, this cell contains just 2 chromosomes, and remember to count the number of centromeres. Because sister chromatids are not considered separate chromosomes, so during meiosis 2, the sister chromatids separate. But each gamete still only contains 2 chromosomes, half as many as a germline cell. So because mitosis and meiosis use similar terminology, it is easy to confuse the two processes. So you can see figure 9.9, which I'm showing you here, that compares the two processes side by side. Both processes start with a diploid cell, but you can see that early during meiosis 1, crossing over occurs, and that that as a consequence, homologous pairs are not individually centromeres that line up at meiosis 1 metaphase plate. So these two differences result in haploid cells in meiosis and diploid cells in mitosis. So let's get to the evolutionary consequences of sex. So you will list the principal mechanisms that generate new genetic combinations in meiosis. Meiosis is a lot more complicated than mitosis. Why has evolution gone to so much trouble? Well, our knowledge of how meiosis and sex evolved is still growing. It is abundantly clear that meiosis and sexual reproduction have an enormous impact on how insects continue to evolve today because their ability to rapidly generate new information. 3 mechanisms which make key contributions: independent assortment, crossing over, and random fertilization. So independent assortment is that reassortment of genetic information or genetic material that takes place during meiosis, and it is a principal factor that has made it possible the evolution of eukaryotic organisms in all of their bewildering diversity over the 1.5 billion years. Sexual reproduction represents an enormous advance in the ability of organisms to generate genetic variability. To understand, recall that most organisms have more than one pair of chromosomes. For example, represented in figure 9.10 at the bottom of the page 162, it has 3 pairs of chromosomes. So I say again, that organism has 3 pairs of chromosomes, each offering offspring 3 homologs, offering each of those 3 homologs from each parent. Purple from the father and green from the mother. The offspring in turn produces gametes, but the distribution of homologs into the gametes is completely random. A gamete could receive all homologs that are patterned for the origin, as on the far left, or it would receive all maternal homologs, as on the far right, or any combination. So independent assortment alone leads to the 8 possible gamete combinations in this example. And in humans, gametes receive 1 homolog for each of the 23 chromosomes. But which homolog of a particular chromosome it receives is determined randomly. So each of the 23 pairs of chromosomes migrates independently. So different possible kinds of gametes that can be produced. So to make this point to his class, one professor offers an A course grade to any student who can write down all the possible combinations of heads and tails, with flipping a coin 23 times. No student has ever won an A. There are over 8 million possibilities. Crossing over. So with crossing over, the DNA exchange that occurs in the arms of non-sister chromatids, crossover adds more recombination to the independent assortment of chromosomes that occurs later in meiosis. Thus, the number of possible genetic combinations can occur among gametes is virtually unlimited. Random fertilization. This is the one I did not mention earlier in this lecture. So, the zygote that forms a new individual by the fusion of 2 gametes produced independently, so fertilization squares the number of possible outcomes. So it'll be 2 to the 23rd times 2 to the 23rd. All right. There are over 70 trillion possible outcomes. So there may be a doppelganger somewhere, but there are differences in everyone on Earth genetically. So the importance of generating diversity. Paradoxically, the evolutionary process is both evolutionarily and conservative. It is revolutionarily in the pace of evolutionary change is quickened by genetic recombination, but much of the results from sexual reproduction. So it is conservative in that the change is not always favored by selection, which may instead preserve existing combinations of genes. So these conservative pressures appear to be greatest in some asexually producing organisms that do not move around freely and that live especially demanding habitats. Invertebrates, on the other hand, the evolutionary premium appears to have been on the versatility of the sexual reproducing as the predominant mode of reproduction. Whatever the forces that led to the sexual reproduction, it is evolutionary consequences that have been profound. So no genetic process generates diversity more quickly. And as you will see in Chapter 10, genetic diversity is a raw material for evolution. It is a fuel that drives it and determines its potential directions. So class, this has been your instructor, Skylar Huff. Please be sure to visit page 164 to put the chapter to work. And again, also, look at the bottom left to retrace the learning path. Thank you all for listening. Prepare well for your test.